the annual Life Sciences Panorama, FACNI's Life Science flagship event. It's my pleasure to welcome you to this webinar. I'm Ludivine Wolczyk, the Executive Director of FACNI, the French-American Chamber of Commerce of New England. This event is organized since 2012 in partnership with our Life Science Forum, co-chaired by Dr. Gigi Shafai, who will be our MC and moderator today, as well as Laure Berliner, who is joining us online. FACNI is a nonprofit association. We are based in Boston and Cambridge at the Cambridge Innovation Center, a spin-off of MIT. We have been in operation for more than 35 years, and we belong to a national network of the FACC. As you can see, we are present all over the United States. FACNI specializes in life sciences and innovation, but many of our chapters have specific capacities. Through our network, we are able to support the launch and the development of French companies all over the United States. We are also able to assist you with your international expansion, thanks to our belonging to an international network of French Chamber of Commerce. We are present in 112 countries at this point. Without further ado, I'm very happy to turn it to Dr. Gigi Shafai, co-chair of the FABS, for the introduction of the program. Thank you.
Thank you, Ludivine. Bonjour, bonsoir, and hello. From wherever you're tuning in. My name is Gigi Shafai. I'm the co-chair of the FABS, French American Biotech Springboard. We're the life science arm of the Chamber of Commerce of New England, which is very active in the life sciences community. So FACNI as a whole, as you heard from Ludivine, is a wonderful organization that helps bridge gaps across France and US, and our team mentors many companies that are hoping to expand to the US or vice versa. And in addition, I also work for TransPerfect as the Vice President of Medical Affairs and Digital Transformation. And over the years, working in life sciences, technology has become even more prevalent and important part of how life sciences organizations operate. And so today, our committee, I want to give a thanks to our committee behind the scenes who helped bring today possible and make today possible. Today, we're going to be talking about leveraging AI to accelerate early R&D and clinical development. Just wanted to set the stage a little bit I urge everyone to ask your questions. We'll be done. I know it's a little bit late in Europe for those who are tuning in. We should be done around 1:15-ish, so in about an hour and 15 minutes. So please stay on. I wanted to take a moment to give a huge thanks to our sponsors, Ipsen and Business France, for today and making today possible. Just for those that are not aware, this is a two-part program. We have our webinar and an in-person event later. And so really wanted to thank our sponsors for making this possible. And also please use the chat feature on the webinar to organically submit your questions, and we will take time to answer as many questions as possible, because AI is a hot topic, isn't it? And we wanted to make it extremely practical for everyone who has joined in, so we answer your questions as well. Today's topic in the webinar is focused on research and development, because AI is there's a lot of innovation happening, and it's a big area for development in life sciences. So we'll be focused on early and clinical stage research and development and the opportunities that lie within AI by our experts here, who are speakers who really have been at the forefront of adoption and the knowledge around where opportunities are and lie. And I also just wanted to make sure that everyone kind of just comes in with an open mind here. We have a lot of different size organizations, and especially younger organizations who are curious but might not have the larger budgets of other organizations. We want you to be able to get that one takeaway or practical key takeaway of where might actually be possible that is cost efficient for your organization or brings operational efficiencies. So keep that in mind. Thank you. So I'm really delighted to have you all here, and we can all go off mute now. But we've begun the exploration of what's possible and how we should be thinking about AI. And now, with our focus on early and clinical research and development, I'm really delighted here to have experts across multiple perspectives here. Dr. Lamont, from a Medidata perspective, she's going to provide a little background about what she's doing, but really coming from your clinical background and merging that into technology and what is possible there. So really delighted to have you speaking today. And Dr. Pascal Descargues from Genoskin, he's the CEO and young company that has actually been able to integrate AI into really cool applications here. So the company itself, if you haven't heard about it, is really interesting. But then how they've been able to apply AI is I'm really excited to have you share with the world. And Dr. Maria Wendt, coming from a Sanofi perspective, big pharma that is at the forefront of technology and AI and how we're able to develop new drugs. I'm really excited to hear from you as well. And so we have the team has prepared actually a slide that we're just to get the conversation going here, wanted to really showcase that what we wanted to offer to our viewers today is really the full scope of from discovery all the way through the commercialization and post-marketing experience that life sciences companies offer. And we start off with drugs, but we've got young companies with Medtech and device backgrounds too. So feel free to share your questions and we can apply best practices and comments across different modalities, therapeutic modalities or diagnostic and prevention kind of approaches too. But when you think about any discovery process and research and development process, we felt we had to cover all the angles because you have to think with the end in mind. And we have the experts here today to talk about those different steps. And when you're creating, let's say, a new compound, all the way from non-clinical research to tests to clinical research to bringing a new product to the market, you have to think, well, what do I already have on the market? What does the world, what are the unmet needs in the world that maybe technology can help advance? So we're going to talk about all of these today, these steps. But I want you to think, so Maria is going to represent the beginning of that story. So when you think of discovery and identifying compounds, and we're all going to speak to everything, but putting a human behind this graph that we're showing, we wanted to showcase the early stage, maybe more of a Maria dialogue. Pascal will come in around some of the non-clinical testing and into clinical translational research. And then Elizabeth for clinical into commercial thinking in the market, because population health is what we're trying to improve the lives of our patients and the information needs for our researchers, but really it's for the patient in mind and population health. So we want to make sure that we're accelerating new therapeutic options that matter. And that's what where we wanted to begin the dialogue. So please, I'll start with you, Maria, and then we'll go to Pascal and then Elizabeth, because let's explain the journey as you see it, what you're working on, and your intro. But in your opening remarks, want to hear the one opportunity for a young company that any company, young company, should adopt when it comes to the thinking of using AI. So I'll start with you, Maria, and we'll get into the juicy content.
Okay, but that was already one big question from the very start. All right, so I'm Maria Wendt. Gigi already mentioned I'm from Sanofi, which is of course the flagship pharma company of France. And also add to my credential, my deep, deep love for France. I want to retire in France. So just an announcement to the companies out there in France. I currently, I'm responsible for our innovation strategy for our, let's say, very novel therapeutic formats. When I say very novel therapeutic formats, it's the protein-based drugs. So these are the biologics. And these are, in fact, the de novo protein design, all this excitement that just won the Nobel Prize recently. And the other aspect, what I look after is, in fact, the innovation in AI as applied to protein-based drug design. So I don't even talk about discovery anymore, because this is exactly what we hope for these new technologies to help us with, that we move from discovering, basically passively, sifting through libraries to look for a beautiful molecule, rather to move into intelligent design. So it's really a very, very exciting time, I would say, for healthcare, for society. But we intend to really utilize all the power of AI for creating these new drugs. And advice for a young company entering this area. I have to say, we recently had to look for a partner, actually multiple partners, as we go into this new exciting space. And there was a lot of companies, of course, that's been recently funded by VCs. I count over 100 companies that are really getting into this game. So it's highly, highly competitive. One big observation is that there has been a lot of focus on building computational platforms to generate a good binder. So a good molecule that would interact, right, with the drug target. And then that is fair and good. That's a good ambition. However, it is not enough. It is important to differentiate yourselves about how to make that binder truly a therapeutic molecule, that it will be effective in the patient, that will be functional, and that it can really be developed. And the other really important advice is something that Odd mentioned earlier, that while companies might be very computationally oriented in terms of their technology, not to forget to have the scientists, let's say the more classical chemists and biologists right next to them. Because it is really when you have those expert chemists and biologists armed with AI, when you make the most of this technology.
Thank you, Maria. Pascal, what are you up to over there at Genoskin?
Thank you, Gigi. So I'm Pascal. I'm the founder and CEO of Genoskin. Genoskin is a small company. We are about 45 people based in Toulouse, in France, and out of Boston in Salem. And what we are doing at Genoskin is we try to help our customers, the biotech and pharmaceutical companies, to really de-risk the development of biotherapies that are injected in human skin. We are focusing on human skin, and we are using actually discarded human skin samples from plastic surgery as an alternative to animal experimentation to help our customer having access into human data even ahead of clinical trials, even before testing in human. And you mentioned, I really like this first presentation where we see the drug development process. And the challenge we try to find solution is this drug development process today is not very efficient. It's more and more expensive. It takes 10 to 15 years. We talk about 1 billion to 2 billion in order to get one molecule on the market. And what is fascinating for us, and also it's our patient, it's the attrition rate. So 90% of everything that is invested in the drug development process is not something that will reach the market, meaning that out of 10 molecules or 10 projects, 9 will just stop before the market. So we have seen that as a big need for the society, for the patient that we can all be in. We develop technologies to bypass the limitation of animal experimentation, for example, that have been used since the last 80 years now. And we use skin models. We obtain a lot of human data, a lot of molecular data. And what is difficult is, even if we get all this beautiful data, it's to be able to draw conclusions for our customers. So here's the product, potentially toxic or not, when injected in human skin. Is it efficient to inhibit inflammation? And we need to be very accurate, to precise. We cannot say just yes, potentially yes, or no, depending on how you analyze the data. So the first use of artificial intelligence for us is definitely the capacity to better analyze all different molecular data we can obtain based on transcriptomic, proteomic analysis and everything. How we can integrate everything, how we can make a summary and really be able to draw conclusion. I would say even actionable conclusion. So our customer can rely on that and then move forward on their own project. This is something very important. We also use, and this is fascinating, we had this beautiful presentation at the beginning showing all the potential of artificial intelligence, but tools just like ChatGPT. I mean, it's really nice. It's so cool. We can do a lot of things, not only for marketing content, but if you need to really summarize the different data to be able to, for example, when you do a report for a customer, to draw a conclusion, to have like something very accurate, this tool, I think, can really help us. The challenge is we are biologists, so we are not like computer scientists, not all, and we are a small company, so we cannot hire like big companies, a bunch of hundreds of people working in this field. So this is what is difficult also. It's to find a mindset or to help people to integrate these tools in their work every day. The second best opportunity we see is we would like to change the way we do drug development. And we think that the data we generate with our system can be used to develop a software algorithm based on artificial intelligence, neuronal network, for example, to be able to really predict what will happen during a clinical development, for example. So not only be able to understand what we see in our system, but to be able to predict a little bit like the weather channel, you know, to understand if at Friday at noon we will have 50% of rain or not. And I think this is a very important step for the drug development. We need to sort out all drugs that have a chance to just do a failure because they are toxic, because they will not be efficient in patient, and we need to do it before the clinical trials because clinical trials are so expensive. So this is something we developed today. It's the idea. It's having tools like biosimulation that go beyond animal models or organ-on-chip models, or even what we develop at Genoskin today.
Thank you, which actually brings us to our segue into the clinic. So Elizabeth, please share, you know, your background, what you're working on, and that opportunity for AI that every company should be thinking about from the clinic.
Excellent. Thank you, Gigi. So my name is Elizabeth Lamont. I'm a medical oncologist and health services researcher, and I am vice president of clinical development at Medidata AI. Medidata is a software company that supports a clinical trial ecosystem, including with one of our big products called RAVE, which is electronic data capture. I would echo exactly what Pascal said, that an AI that can accelerate this arc that you showed so nicely, that starts with an idea for a drug to improving population health, that takes a long time currently, and it's very expensive. And I see AI really accelerating that in a variety of ways. It was great to hear about Pascal's ways that it's accelerating the preclinical space. And I know it's accelerating the in silico space, and Maria, I think, is going to talk about that. Where I see it accelerating development is in the sort of the clinical investigation, clinical trial space. Historically, clinical trials are sequenced from what's called phase 1, which is when you have a new compound and you're trying to test and see what the maximally tolerated dose is in a human. Then the drug, once the dose is understood, it's studied in a phase 2 setting, which is often a single-arm trial, and you're just trying to see, does the tumor shrink or not, for example, in oncology. And then if it shrinks, then people move to the phase 3 setting, where they compare this candidate drug to a standard of care. And these are usually very big trials, very, very expensive, many, many years. And sadly, only half the time the new drug is shown to be better. So the idea is there a way that we can get information at an earlier phase? And so what we did was we helped a company who had phase 1 data, and they thought they had a good outcome in terms of survival. But it's sort of like, well, compared to what, right? So they had 18 patients who were treated with a regimen for ovarian cancer, and they saw a certain progression-free survival, a survival endpoint that looked historically better than most. What we were able to do was we were able to help them to create a matched synthetic control. We call it synthetic control arm. It's an external control arm. So we were able to match patients from historic trial data to the small number of patients in the phase 1 setting. There were 18 patients in the phase 1 trial. We were able to match 15 of those with historic patients who received a standard of care, and then estimate the outcome of interest. And it turned out that people were right. The company was right to think they had a good drug, because the progression-free survival was 50% better in the new drug. And so that did a couple things. One, it helped the company to say, yes, let's go forward and develop this drug. So it's a phase 1 setting. It's really early. There are only 18 patients, and yet there's a comparative effectiveness estimate that was promising. And number two, with the effect size that we were able to estimate, they were able to right size their subsequent randomized trial, and they saved 20 patients, it turned out, because the effect size was so big. When you think about how much it costs, it's about $100,000 per patient on a trial. So if you save 20 patients, maybe that's close to $2 million saved. And so I see, the clinical space is also being able to benefit from AI and accelerating the timeline.
That's a wonderful example. And I think one of the pieces there was around how, if you apply it in a phase 1 time frame, it gets to Pascal's point about de-risking a program and knowing information up front that changes the landscape. What if that same exercise told you that it would work better in a subpopulation that might help you design the next stage trial? Then you're focused on, these patients are actually giving up time out of their lives and their the stress that they have with their conditions to participate in studies. So if there's the operational and cost efficiencies to organizations that you're alluding to, but to save those patients, you're also saying is also saving their time and to get the right patient in the right trial too. And so love this discussion. I'm going to pivot to you, Maria, here on the when we talked originally, we had in our preps, I loved when you described systems biology. So I'm going to go back to outside of the clinic and to the roots, because with the clinic in mind, tell us, please explain systems biology to the non-biologist.
Okay, I will try. But before that, there was actually some really important nugget that Elizabeth shared with us. Saving 20 patients could be equivalent to saving $2 million. Did I get that number right, Elizabeth? Okay.
Yes, you did. That's an estimate, but yes,
that's an
thank you.
estimate, but it's showing you the magnitude of the potential impact when you really try to improve on that end of the R&D value chain in saving patients and saving on the clinical trials. Because there are many companies that are trying to help us generate new molecules, but the savings in the generation of the molecules, let's say the timeline savings and maybe the number of molecules that need to be tested will be less, that it's still actually quite smaller than the savings that you can get from applying AI into the development part of the value chain. The magnitudes are simply different, although one still really needs to apply AI in as much as possible, in as much of the value chain as possible, because anywhere that we can optimize in the value chain will be very, very helpful. But there is really, to me, a particular important, big, urgent opportunity to really apply AI as much as possible to get to the right patients, to get the patients in there, and to, of course, make sure that the drugs will work best in the patients. That's also why I'm also a really big fan of what Pascal's company does in helping that. Now, to the systems biology, yes. So systems biology, I was sharing with Gigi before that when we think about drug discovery and basically work in pharmaceutical industry, there has always been this paradigm of one drug to one target, one target, one disease. Like if you have a pathway that explains the biology of a disease, that if you can interrupt even just one target, that you will in fact interrupt and hopefully help that particular pathway and therefore that disease. But we now know that that's not true, that when you have a biological pathway, you interrupt one part of it, there are many other ways that the pathway will be compensated on. And therefore, it's important probably to interrupt the pathway in multiple points. And that is when we start entering systems biology, when you want to look at things holistically, not one target, but multiple targets, and also at different dimensions, whether it's transcriptomics, the proteins, or the metabonomics. And back then, maybe people think of the one target, one pathway, one disease. It's because they're limited by the types of technologies that they have back then, the expense of the experiments, the difficulty, the throughput. But today, there's no more excuse because the instrumentation is just so sophisticated. The readouts will be deep, not shallow. We have genomics, the omics that Elizabeth mentioned, and also Pascal mentioned, not to mention all this multiplexed microscopy, fluoroscopy, that you simply have so much rich data that you can take now on those various systems dimension. Not to mention now the AI, the math that is available to do that. And so right now, there's no more excuse not to really target diseases in the systems way. And this is also, in fact, what I think what would be so special opportunity with AI is that we get to design more complicated drugs for the more complicated biology that we now wish to target. And this is, in fact, the situation at Sanofi. We think that, of course, AI will help accelerate drug discovery, but even better, that AI will help us make much, much better and smarter medicines.
Thank you. And so I encourage people to start putting questions in the chat because we are almost up on time as far as the discussion goes, believe it or not. But while we do that, Pascal, I'm going to pivot to you on an example that you can think of that where you tested like screened molecules or something. Like, how did you do? I want people to be able to visualize an example of what you've been able to, like a decision point on the de-risking that you've mentioned that's possible, because you've screened multiple molecules, I understand, and maybe a safety example or I think it's really cool how you can test skin and human tissue and find information.
Yeah, it's the main project we have around AI. We call it ID Atlas. And what we do is we inject in our skin models, in our platform, we inject drugs that are commercially available. You can find everywhere that are biologics, vaccines, whatever. And our system is a human skin organ. It's immunocompetent, meaning that you have like about 1 million T cells, for example, per square centimeter. You have a total of 17 different type of immune cells, plus the keratinocytes, plus the fibroblasts, plus the blood vessels, plus the nerves, plus a lot of, it's a complex environment. It's a teeny small biopsy. It's already super complex. And what we do, we mention omics. So we get, after the treatment, we obtain, we sample the tissue and we do a transcriptomic. We look at the single cell level. So if you consider the bunch of 500K cells, we look at about 20,000 of genes in 500,000 of cells. It's a lot of data. You can do the same at the protein level, so proteomics. And you have all this data. What we do today, we built our own biosimulation system using a neuronal network, and we actually mix all the data we have to the clinical outcomes that are known for these drugs. And this idea is to make connection between what you can see in our system in vitro in the lab, and what is the real clinical outcome for this drug, in particular regarding injection site reaction. We have a huge interest in injection site reactions. And the idea for us is really to be able to test a new drug, a non-known drug, a new molecule that is, you don't know anything about clinical trials, so it's a very new, and be able to connect when, based on the molecular footprint obtained in our system, to connect to the clinical outcome, the potential or the prediction of the clinical outcome for this particular new molecule. So this is how it works. And I think AI is the tool that helps us, because Maria, you mentioned the equipment, the instrument that has totally changed life science because of this omics, all these capabilities. We can get a lot of data. And then how can you use the data? I mean, it's too much, right? You cannot put this in the brain of a researcher and something will come out. So the hope is in silico, artificial intelligence will help us to use all this data, to connect it, to see a trend, to be able to connect to, for example, for us, injection site reaction, clinical outcomes for injectable drugs in the skin. So this is something that is ongoing, and we don't know if we will be successful or not. I mean, so far we don't know. So we hope that the AI will still continue to improve, so we can use tools, we can incorporate updated tools to be able to get a precise reply, a precise answer for our quest.
Thank you. And last question for Elizabeth before we move to Q&A. But I wanted to, going back into the clinic here, I think one of the coolest areas with AI is to be able to identify, stratify subpopulations. And I'm just curious if you could shed light on applications of AI or examples that maybe have helped the subpopulation conversation. And the reason I say this is because I know, a lot of clinical trials, they have all like list of exclusion criteria, and they, kids can't be in this study, and the really complex patient with a certain scenario. Like, have you dabbled into AI there, and how we can really help bring therapeutic options with data in those patients maybe we might have not been able to study before?
Gigi, that's a fabulous question. And one of the things we have is a tool called Intelligent Trials. And what that does is looks at prior data on enrollment of patients into different sites. And you can get some metrics from that. You can understand which sites maybe have the lowest dropout of patients. You can see which sites are enrolling patients whose demographics are perhaps representative of the US. Unlike most trial patients have demographics that are very different from the US. They tend to be white, young, healthy, and that's a real bias, and it's a real problem. So FDA, the government, lots of groups are coming together to say, we need better representation of Americans in trials that are leading to development of new drugs. And we have been able to help that through helping companies understand, which sites are able to enroll older patients more often, which sites are able to enroll people of color. And that has been a huge, exciting advance. And that's something we're really happy to keep working on, because until these big trials are representative of the US, it's hard to know how to treat the patients who aren't in those samples. So I think that's a really exciting way that AI is making the world a better place.
Wonderful. Thank you. And that's an area which led me into digital health, honestly, because to reach the patients, to communicate with the right patients, to provide treatments to the right populations. I think that's what we're all trying to work on in our own ways, whether from discovery through the clinic and even post-marketing type work that we're doing. So thank you all for the discussion. And I'm also not seeing a whole lot of questions, but I promised we'd go back to the fibrosis question, especially, I think, Maria, that's more directed to you. And are we able to use AI to mimic what mutated protein can induce as an effect, like a fibrosis, and that connection between protein to fibrosis and impacting it? What can we learn from that?
Yes. So in fact, my answer will be helpful to this question, but for probably many other questions that came up. And that is, if you have the data, yes. What I mean is, this is about, the question is about prediction. Of course, there's the protein-protein interaction prediction, but then there's interactions that are really, in fact, relevant to fibrosis. If you have the wealth of data in the background that allows you to create a predictive model, yes, I think there's no reason why AI will not be able to do it. But again, it's predicated on the availability of sufficient data, something that Pascal actually repeatedly drove on in this discussion today. And I have to say, there's also this question about immunogenicity, because our fantasies really about AI and protein designs. In fact, we have this beautiful de novo designed proteins never seen or experienced by any being before. And therefore would be at high risk of potential immunogenic reactions and therefore potential failure to the clinic. So really, really important question. And in fact, we have a challenge there because we cannot predict that, because there's hardly any data, very, very small amount of data of immunogenicity with these types of molecules. And because of that, we have to then rely on more in vitro type of assays to predict immunogenicity. So that high bar will remain there because it's really about patient safety. But the best that can really be done right now, there are in silico tools, but it's really important to use some in vitro assays. At Sanofi, we use 2 different in vitro assays for immunogenicity prediction.
Thank you. Pascal, we've got a question for you. Do you think that the predictive models you're developing could be developed in many other fields? What would be the impact on animal model testing? And is the FDA ready to accept IND data with little animal data?
So I think, yes, we can develop similar approaches for different fields. It's called biosimulation. And there are some people who are trying to do a digital twin of a patient, but if you come back to the to non-clinical testing, it's a digital twin of PK/PD, it's a digital twin of sensitization, digital twin for toxicity for certain organs, for example. So perhaps the best approach is to do little by little. I'm not sure we'll find just one algorithm that will reply to everything. Not today, I think. And to reply also to the question regarding the FDA. So I'm not the FDA, so I have no idea. But I mean, they open up recently, less than 2 years ago, they open up the system to the use of non-animal data, thanks to the FDA Modernization Act, So now it's in progress since the end of 2022. And I think the idea is really to be able to find a way to have more data that can be translated to the patient. So this is coming, and my my thoughts are that in 30 years, in 50 years, yes, probably we'll have little animal data and more in silico, more in vitro, more in tubo data to do drug development, and this probably will also impact clinical development, perhaps with less patient and more AI, like Elizabeth said. So yeah, the change is coming. It's now.
So to piggyback off of that, there was a question: do you envision the use of AI to be regulated in the future? And I think Pascal, you mentioned the Modernization Act when we discussed too, but and there's different applications of AI. So I know that, like, for example, EU has a recent regulation that came across. It's like the first regulation for AI that came through, and there's definitely work to be coming. But from a R&D perspective, do you want just piggyback off of your comment, Pascal, there?
Yeah, and from the EU regulation, I'm not sure this regulation is in favor of innovation, but this is another story with Europe or European countries. But you know, it's very interesting when you look at the guidelines for, you know, when you go to the FDA. I mean, there is nothing in the text on the guidelines saying that you have to use this particular method.
Right.
So my understanding is the sponsors, they need to select a method to screen the drug, to test for toxicity, for efficacy, and they come to the regulator with data, with you know, things that can help to take a decision. But there is, so it's why the change is very interesting with the FDA Modernization Act. It's like it's an opening, because before everything was called preclinical, now it's non-clinical, and before everything was called animal data, and now it's open to in vitro, to human cell, to organoids, to in silico. So it's larger now. There are more room for innovation, for competition. If you think two similar files for similar drugs that target a similar disease, for example, one is coming with only animal data, very classic old way, and another one has in silico data based on use, or also in vitro models with human cells or whatever. So what file would be the most convincing? This is the question. I think the idea was to open to more competition.
Right. So I think just also to recap this too, is that, you know, to ensure responsible AI also is dependent on the disease you're trying to treat. And so maybe they are not being as prescriptive, because if you're dealing with proteins and immune systems, and you have to do what's right for as a sponsor, for the population and the target that you know you're working on. And so to leverage technology where it needs to be, but to also know where that human oversight will help inform the technology, is a squishy thing, right? And so that there may be factors into what future regulations are prescriptive about or not prescriptive about, and we'll see what happens, but we know that there's so many applications for technology here. In our last, because we are running up on time, but Maria, maybe you could cover the foundational language models. and so, because there was a question earlier about predictive models, I believe.
Okay, thank you, Gigi. Because yeah, we understood that many companies, young companies today, a lot of their foundation, well, their platforms are based on certain foundational models. So we thought we'll comment about that quickly. So my feeling is the jury is still out. So there are, of course, many different types of foundational language models. In our case, in drug discovery, you are seeing a lot of protein language models out there getting published. So my answer there is it depends on what modality. So that was also part of the question you are working with. Are you working on antibodies or general proteins like peptides or cytokines? Because there are foundational models that are built on all proteins, but there are also now newer foundational models that are specifically for a particular modality, like specifically for antibodies or single chain antibodies. And I say the jury is out because foundational models, as you know, it's just a means of translating a protein sequence into numbers so that you can operate on them mathematically. So it's really a question of which set of numbers represent the protein best. And you will only see that when you use the language model on a downstream task, let's say predicting expression, predicting binding, or predicting thermostability, or predicting immunogenicity. And there has not been a rigorous comparison yet of how each one of these language models do in each one of those different prediction tasks. Okay, so the jury is still out. However, I can just share with you that related to immunogenicity, we did see that certain language models predict immunogenicity better than others, and specifically for antibodies. So there's some excitement there worth looking more closely into.
Thank you so much, Maria. Thank you all for this discussion. We could probably spend hours talking about all the potential use cases for AI or the ones that you've already adopted or the potential ones coming down. It's interesting to see where the field will be headed. And we hope that everybody has learned something new or has taken a nugget from our discussion and our presentations today. I think just to close our programming for today, just keep an open mind around AI. If you are you joined, you're curious for a reason, maybe you haven't used it yet, but as you can see across the entire process of research and development, whether you're working on a drug or trying to find something to make your research process internally more efficient, or just de-risk your program and find solutions to bring value to your teams. These are some our discussion, I hope, gave you some ideas as to where you could do it on the early stage or on the later stage, or even just inside. Pascal brought up how you could research the existing information and even create content, and to be able to summarize everything that you learned from your traditional research approach might be the way you feel comfortable in adopting AI. So it's just food for thought, and we hope you learned something and really got introduced to some experts that are in our area. And Boston has a lot to offer to the world. And you heard from four doctors in the area who are tackling it from different angles. So I hope that everybody enjoyed, and thank you for those that stayed online. And whether you are wrapping up your day or finishing up your lunch, bon après-midi, bonne nuit. And we look forward to tonight's part two program to continue the dialogue on the impact of AI in life sciences. So thank you all.
Thank you, everyone.
Thank you, everybody. Bye.